started, are you?
Yes.
You ready? And everybody has this packet, right?
Yeah.
Okay. So hopefully you guys had worked on this over the weekend before coming to the session, so that mostly you're going to be checking your answers or asking me maybe why an answer that you had recorded is not the correct answer, something like that. Okay? So feel free to ask questions when you need to. Okay? All right. So basically this first exam is going to cover chapters 1, 2, 3, and 4, right? So you'll see that in this packet, you know, the topics are all kind of mixed up because that's how you're going to see it in the test. You're not going to see just chapter 1 questions, chapter 2 questions, chapter 3 questions, chapter 4, okay? You just, you know, you're going to get to see them and answer them according to what you've learned so far, okay? All right, so first of all, enzymes. Enzymes is the first topic, and all I'm asking in this question is just, what do you know about enzymes? Can you tell me 3 different things about enzymes?
Digestion.
Okay. Well, there are specific types of enzymes that help to aid digestion. Okay, but what about just more general? What type of a biological molecule are enzymes? Are they lipids, nucleic acids, proteins, or carbohydrates? Okay, we've got some conflicting information there. What are they? They are proteins. Okay, so enzymes, number 1, they are proteins. Enzymes are proteins. What is their function? What do they do in a chemical reaction? Okay, well do they make it slower or do they make it faster?
Faster.
They make it faster. So enzymes speed up a chemical reaction by doing what? They do something with the activation energy. That's important. What
ATP
So raising or lowering the activation energy. How do enzymes speed up a chemical reaction? By raising the activation energy or by lowering the activation energy? Lowering. Okay? So enzymes speed up a chemical reaction by lowering the activation energy. Okay. What do enzymes achieve because they have that tertiary structure? Remember the levels of protein structure? What makes all enzymes different? They have a unique what? A unique shape, a unique 3-dimensional shape. Okay? They have a unique 3D shape.
tertiary.
Yeah, this type of a protein and enzyme achieves that unique 3D shape when it reaches the tertiary structure. Okay? So the unique 3D shape, specifically of what part of the enzyme? There's a specific part of that enzyme that has a really important shape. What's that called? You guys remember? It's the site where a substrate will bond with the enzyme. Remember what it's called? Unique 3D shape of the one site, right? The active site. Okay, the active site. Unique 3D shape of active site. Right? So the active site, that's important. The active site, it's kind of shaped like a puzzle piece. Okay? And so the active site of the enzyme is going to bond with a substrate. So maybe the substrate is the disaccharide sucrose. Okay? So the sucrose molecule has a shape that is complementary to the shape of that enzyme's active site that it can fit with. Okay? And then that enzyme can help to break the disaccharide sucrose into glucose and fructose, for example. Okay? Now, keeping that shape is absolutely essential for the function of that enzyme, right? So what different environmental factors can cause an enzyme to lose its shape? Extreme what? Heat. Extreme heat and extreme
Cold.
not cold. pH. Remember pH? Extreme changes in pH. Okay? So extreme heat or pH You guys know the word? What happens when the enzyme unravels and loses its shape? Do you know what that special word is called? It's the word that starts with D. Denature. Denature. When it denatures, it unravels and it loses shape. And it's not going to function anymore. Okay? So that's 4 different things about enzymes that I've listed, right? Enzymes are proteins. They function as catalysts. You could even say catalysts because catalysts are responsible for speeding up a chemical reaction. And enzymes do this by lowering the activation energy, or the energy of activation. Enzymes have a unique 3-dimensional shape, specifically of that active site, that determines what substrate is going to fit with it. And if an enzyme is exposed to extreme heat or extreme pH, okay, because most enzymes function best at neutral, then it will unravel and lose its shape. It will denature, and it's not going to function. Okay? So that's a lot of different things about enzymes, right? So you can always give me some of that if you saw an extra credit question like that on the test, or at least identify some of those factors about enzymes in the multiple choice question. Okay? All right, so that's enzymes. How about the cell theory? Do you guys remember the cell theory? There's really 2 parts to the cell theory. So what is the cell theory? You want to start it?
All living things are composed from cells, and all cells form all other cells.
That's right, okay? All living things are composed of cells. That's the first part. And, say it again, one more time.
All cells form all
There you
other
go.
cells.
And all cells come from other cells. Okay. What was the technological advancement that led to the development of the cell theory? The microscope. Good. Okay. Definitely the microscope used for looking at very small things. Okay. I have had a student before tell me on a question like that, if I gave that as the extra credit question, the telescope. Okay, please don't tell me telescopes. Telescopes are used for looking at things very, very far away. Okay, the microscope is used for looking at things like cells that are microscopic.
Anton
van Leeuwenhoek is the person who went down in history as the most famous microscopist because he designed a modern precursor to the microscopes we use now. Okay, he discovered sperm cells, bacterial cells, and red blood cells. Okay. All right, so now we're going to go through a little bit of multiple choice here. So matching the organelles and structures with their function, right? So I'm going to read the sentences, and you guys together should give me the answer, not the letter. Okay, give me the name of the organelle. That's better practice. Okay. So for number 1, I'm a complex of DNA and proteins that makes up the cell's chromosomes. Chromatin. Very good. So chromatin, that's letter C. The answer for number 1, letter C. Chromatin. Number 2, I make membranes and transport products.
Rough endoplasmic reticulum.
Yep, the rough ER, the rough endoplasmic reticulum. Letter F for number 2. Number 3, I'm a double membrane with pores, and I'm attached to the endoplasmic reticulum. The nuclear envelope. Letter D. Letter D, nuclear envelope for number 3. Number 4, I carry products from the endoplasmic reticulum to the Golgi apparatus. Transport vesicles. Letter H for number 4. Number 5, I'm the control center of the cell, and I contain the cell's chromosomes. The nucleus, right? Letter A. Nobody should miss that one. The control center of the cell. Gotta be the nucleus. Number 6, I make proteins for use in the cell, and I am synthesized in the nucleolus. That's right, ribosomes. Ribosomes, letter B for number 6. Number 7, I expel excess water to prevent a cell from bursting.
Contractile vacuole.
Contractile vacuole, letter G. mean, letter J, letter J. Contractile vacuole, letter J. Number 8, I contain digestive enzymes that break down and recycle damaged cell parts. Lysosomes, letter I, lysosomes. Number 9, I manufacture a variety of lipids, including oils, steroids, and phospholipids. The smoothie R, right, the smooth endoplasmic reticulum. Letter E for number 9. And number 10, I package, right, hint, 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 sort and ship cell products produced by the endoplasmic reticulum.
Oh, yeah.
Everyone got that one. That was even easier than the nucleus. The post office, right? Yep, so Golgi apparatus, letter G for number 10. Packages, sorts, and ships. That's got to be Golgi. Okay, so moving on to the back of that page, we've got some questions about bonding. Okay, so what kind of a bond forms between water molecules? Number 5. Hydrogen bonds. Good. Hydrogen. Hydrogen bonds. And number 6, the type of bond that forms between amino acids. My students who were in lab this morning should not miss that, right? We talked about that. The type of bond that between amino acids is what? Peptide bonds. Peptide bond is a special bond that links amino acids. And then number 7, the type of bond that forms between nitrogen bases and DNA. What's the type of bond that forms between the nitrogen bases and a molecule of DNA holding the two spiral staircases, the two strands together?
Carboxyl.
What kind of a bond, guys? So remember, when we looked at the picture in the slide, we saw the double helix. Okay. And we saw the nitrogen bases extending from the strand, and then there was a bond represented by like 3 little dots, and that's the bond that holds the nitrogen bases together. Okay, nobody knows? What kind of a bond links together the nitrogen bases and a strand of DNA? Hydrogen again. Hydrogen bonds. Okay, so it's a hydrogen bond one more time here. Hydrogen bonds form between the nitrogen bases. Okay, hydrogen bonds are very important. We're going to encounter them a lot throughout the semester. Okay, so hydrogen bonds form between water molecules. Hydrogen bonds form between nitrogen bases. Okay, remember that it's hydrogen bonds that cause the secondary structure of protein formations. So hydrogen bonds are going to be found to be important throughout the term. Okay. Okay, so number 7 is hydrogen bonds. How about number 8? What are amino acids? Yeah, so they're what about proteins? They're not just proteins on their own, but they are the building blocks of proteins. Good. Okay. We just get some clear space here on the board. All right, so amino acids are the building blocks of proteins. The next question is, what 2 functional groups are found in all amino acids?
Amino group and carboxyl.
An amino group and a carboxyl group. So just to make sure that you guys can see this, we'll just draw out the structure of this amino acid that we have here. Okay. So we talked about functional groups. Remember that there was a whole slide where you saw a whole bunch of functional groups. And so you guys just told me that one of them was the amino group, right? So which one was the amino group? Is it this one? No, right? It's this one, correct? That's the amino group. All right, so amino group. And what's this one? presence of the amino group bound to the central carbon atom, and then that central carbon atom also bound to a carboxyl group. Okay? Do you guys remember what makes all amino acids different? Well, how are there 20 different amino acids? Because the R group, and this is an example of an R group right here. Okay? But they are the same, all based on the presence of the amino group and the carboxyl group. And there are 20 different R groups, which is what makes them different. Okay?
Does all need anything specific?
No, it's just like kind of like a holding space, kind of like how the word, the letter X in math problems, right? X is just a variable that changes. So sometimes you can also call even the side group. It's called a side group or an R group. It's just a general catch-all term for something that changes. Okay. Okay. How about number 9? How do the nucleic acids DNA and ATP differ in function and structure? Okay. Well, let's first talk about function. So the nucleic acid DNA, what is that involved in? What's the whole purpose of DNA? What is encoded there? Your genetic information. So all of your hereditary information is encoded in your DNA. Okay, so here's my rough sketch of DNA, and it's all about heredity. It's all about hereditary information. That's its function. Whereas ATP has what function?
Energy.
Yeah, energy, fuel. Okay. So very different functions, okay, between DNA and ATP, but they're both nucleic acids. So let's talk about how they differ in structure and function. It might be helpful to sort of kind of map out what their structure consists of, and then we can point out the differences, okay? So for ATP, what are its building blocks? Okay, what is it going to have? Okay, it's going to have adenine, which is a nitrogen base. Okay, so it's got a nitrogen base, which is adenine. That's actually what gives it its name. ATP stands for adenosine triphosphate. Okay, so what else? It's got one nitrogen base, adenine. What else does it have? It has 3 phosphate groups. Okay, triphosphate, 3 phosphate functional groups. Okay, what else does it have? One sugar group. And what is the sugar group? What is it? I heard it, Dory. Ribose. Ribose sugar. Okay. So ATP only has one sugar group, and it's ribose. It has 3 exact phosphate groups, and it has 1 nitrogen base, adenine. Okay? Now what about DNA, though? Let me take rid of this label here. So this is a DNA molecule right here. What are its building blocks? Does it have sugar groups?
Yes.
Yes. Okay. Does it have a certain number of sugar groups? A certain number of sugar groups? No. It has a whole lot of sugar groups. There are sugar groups that constantly repeat along each strand. So it doesn't have necessarily a definite number of sugar groups. Okay. It just has sugar groups. And what is the sugar group in DNA? What is it? Let's say that together, okay? First of all, DNA stands for deoxyribonucleic acid, okay? So what is the sugar called? Deoxyribose. Let's say that together. Already, one more time. 1, 2, 3. Deoxyribose. Very good. Deoxyribose. Can you spell it too? You won't be counted off for spelling. Don't worry about that. Okay. But you got sugar groups, and it's got to be deoxyribose. What else have you got? Do you have phosphate groups?
Yes.
Do you have a certain number of phosphate groups? You have sugar groups and phosphate groups that constantly repeat along the sugar phosphate backbone of both strands. You've got a lot of phosphate groups. So you can just say phosphate groups. Okay? And then what about the nitrogen bases? How many different nitrogen bases are there in DNA? Okay, there's 4 different types, right? Okay, and they extend from the backbone. So what are the nitrogen bases?
Adenine,
Amine.
cytosine, and
Okay, so A stands for adenine.
Is your cell in it too?
T stands for thymine. C stands for cytosine.
1G.
And G stands for guanine. Okay, you're asking about uracil. Uracil is only in RNA. Uracil is a nitrogen base in RNA only, okay? And we're going to differentiate that in question number 10, okay, that we're going to get to next. All right? So let's now look at the differences for this question. So the difference is definitely function, right? They differ very greatly in function. DNA is for hereditary information. ATP is for fuel. In terms of structure, they have the same basic building blocks, right? Sugar, phosphate, nitrogen bases. But there are some differences. So for ATP, the sugar group is ribose, whereas in DNA, the sugar is deoxyribose. Okay, so that's a definite difference. Okay? In ATP, there are only 3 phosphate groups. In DNA, there are many phosphate groups. In ATP, there's only 1 nitrogen base, and it's adenine. Okay? In DNA, there are 4 different kinds of nitrogen bases: adenine, thymine, cytosine, and guanine. And there's many of them. They constantly extend from each of the sugar phosphate backbones, each of the strands of the DNA double helix. Okay? So question number 10 is very similar, okay? But now we're going to throw RNA into the mix as well, okay? And we're going to talk about the common building blocks. And of course, all nucleic acids have sugar, phosphate, and nitrogen base, okay? But what are the differences even within there? So I'm just going to get rid of our protein stuff right here so we can have a nice space to put RNA. So RNA structure is that it also has sugar groups. What is the sugar group in RNA? Ribose. Good. Same sugar that was in ATP. Okay? And that's because RNA stands for what? You guys know? Ribonucleic acid. Very good. Ribonucleic acid. Now, RNA also has phosphate groups, just like DNA does, and they repeat with the sugar groups along the sugar phosphate backbone. Okay. And what about the nitrogen bases? You also have 4. What are the 4 nitrogen bases in RNA?
Adenine, uracil, cytosine, guanine.
Just, yes, cytosine and guanine. Okay, so adenine, uracil, cytosine, and guanine. AUCG.
Are all 3 used for nucleic acid?
All of these are nucleic acids. These are the only nucleic acids that you guys are responsible for. Okay? And all of this is something you're also going to have to know when we get into our 3rd major unit of stuff for exam 3. You'll need to know all this again for chapter 10 content, so you might as well start learning it now. It'll make those extra things that we're going to learn about in chapter 10 easier, okay, to understand these differences, okay? So RNA has ribose sugar groups, it has phosphate groups, and it has the nitrogen bases A, U, C, and G. Just another difference between DNA and RNA is that RNA is only single-stranded, okay? It's just one single strand. And so you'll have ribose phosphate, ribose phosphate, all the way down the backbone, and then extending from the backbone, you will have your nitrogen bases, like A, U, C, and G. A, U, C, G. RNA is single-stranded, DNA is double-stranded, and ATP is not stranded at all. It's not a stranded molecule. And that's largely because it has a completely different function. It's for energy, fuel. It's not for carrying your genetic information or transmitting your genetic information. Yes?
So A and U can only bond together, and C and G can only?
Yes. And that's only going to happen, though, in the process of transcription, okay, which you're going to get to in chapter 10, okay? All right. So the common building blocks between all three of these is sugar, phosphate, and nitrogen bases, right? Okay. And there are some differences within that, you know. The differences with the fact that RNA is a cell that does not have thymine. That's what DNA has. Okay. So it really helps guys to kind of map it out like that to really visualize those differences and commonalities. Okay? All right, so the difference between saturated and unsaturated fats. So we talked about like 3 or 4 different things about those, how they're differentiated, what their structure is. Is that a good marker? All right, so saturated versus unsaturated fats. Okay. So in terms of their chemical structure, okay, what are some of the important differences? Saturated fat is saturated with what? It's saturated with hydrogen, okay? So that means that a saturated fat has a maximum number, so use the number symbol there, of hydrogen atoms around the carbon skeleton, okay? What about an unsaturated fat? Does it have the maximum number? No, that's less than the maximum number. Okay. So a saturated fat, what kinds of bonds? All single bonds, all single covalent bonds are linking all of those hydrogens and carbons together, okay? So you can say all single bonds. What about the unsaturated fat? Is that the same situation there?
No, one or more double.
One or more double covalent bonds. But that's not what we see when we look at a fatty substance, right? We only see its consistency at room temperature, okay? So which one of these is a liquid at room temperature? An unsaturated fat. Okay, so liquid at room temperature. Whereas a saturated fat is going to be solid, right? Like a stick of butter. A stick of butter is animal fat. And so at room temperature, it's nice and solid, right? So like if you had a stick of butter and you unwrapped it, you left it overnight on your counter, it's going to be soft to the touch, but it'll still keep that rectangular shape. Okay? So saturated fats are solid at room temperature, unsaturated fats are liquid at room temperature. Okay? So how about the polysaccharides? Ready to go over the polysaccharides? Okay. Okay, so we discussed 4 different polysaccharides that you guys are going to be responsible for. So let's list them out. Well, we'll just ask you guys, how about this? Which one is the energy storage polysaccharide in animals? Energy storage in animals, like glycogen. Glycogen, okay? So glycogen is only in animals. Okay, and it's for energy storage. Plants have an energy storage polysaccharide as well, right? What is it? Starch. Starch is the energy storage polysaccharide in plants. Okay? The other two that you learned about are for providing structural support. So, which one? Okay, what about cellulose? What is it? What is it? Structural support for what? Animals or plants? Plants. Yep. So cellulose is for structural support. Structural support in plant. Let me just put a line down here to separate it. Okay? And what part of the cell is made of cellulose? The cell wall. Okay? Cell wall is made of cellulose. Now, organisms with an internal body skeleton have a skeleton, and calcium is there to provide strength and support, okay? But what about animals that don't have an internal body skeleton? Animals like lobsters and shrimp that have an exoskeleton. That exoskeleton is largely a polysaccharide called what? Yeah, chitin. Okay, structural support in animals. Do you guys remember what we actually make into dissolvable stitches? Dissolvable stitches are made of chitin largely. That's why they dissolve. Okay.
Is that only an exoskeleton for the
And insects, any sort of organism that doesn't have an internal body skeleton would need to have chitin for that support. Okay. So just try to divide it up like that, guys. Okay, so which of these polysaccharides is in plants? Which of these is in animals? And then what's their function, right? 2 of them are for energy storage, 2 of them are for structural support. Okay, so you just gotta get that straight. Okay, doing okay so far? Yeah. Okay. All right, so now we're done with page 1. Hooray, moving on. Okay, so next page, and the first question there is, what is the difference between an element and a compound? So, let's talk about that. So an element, can an element be broken down into anything else?
No.
No, right? So what is an element? How would you define it?
It's a substance that cannot be broken.
Okay, so it's the purest form of a substance and it cannot be broken down. Purest form of a substance cannot be broken down. Okay? Whereas a compound, what is a compound made out of?
Consists of 2 or more different elements.
Yeah. 2 or more elements bonded together. And can it be broken down?
Yes.
Yes. I know it doesn't say this on the worksheet, but give me an example. Give me an example of an element, a pure element. What? Iron. Okay, iron, carbon. All of those are pure elements. Okay? Give me an example of a compound. H2O, the most common compound on Earth. That's a good one. Example, H2O. What else?
Carbon dioxide.
Sure, carbon dioxide. That works. Sugar. You remember the formula? C6H12. You're in. And then O6. Sugar, glucose, C6H12O6. Those are all compounds. abundant elements in living things. Do you remember that?
Carbon, hydrogen,
Hydrogen's one. What else?
carbon,
Carbon. Yep. What else?
oxygen,
Yep. And what else? Nitrogen. There you go. Big four. Okay. So carbon, hydrogen, oxygen, nitrogen. The four elements that make up the vast majority of living things, right? Like our bodies are 96% just those four elements. Carbon, hydrogen, oxygen, and nitrogen. You guys learned all about trace elements, right? In your assignment? Okay? So, what are trace elements? They're what? Essential elements
minute quantities.
in minute quantities. What else? Yeah, they come in very, very small, minute quantities. Okay, very good. So trace elements, which are not these are not trace elements. These are big, major elements in our bodies. Okay, but trace elements are elements that are found in tiny, okay, and by tiny, it's less than, 0.01%, very tiny, less than 0.01% concentrations in living things. But can we live without them?
No.
Are they essential? Yes. We need them for proper health and functioning, right? For health. Okay. So trace elements are elements that are found in very small, tiny concentrations, less than 0.01%. Even though they're in our bodies in such a small concentration, they are very important for proper health and functioning. Okay? So what are some of the examples that we went in class? When we went over class? So we went over iron, right? And we also talked about what else? Something else that starts with I, right? Iodine. Okay? So why is iron important? Do you guys remember? It helps to transport oxygen through the blood. Okay? And why is iodine important? What's it important for? The health of a gland right here. Your thyroid gland. Okay? Because if you don't have enough iodine in your diet, what happens to your thyroid gland? It swells up and it becomes a goiter. Goiter, a goiter forms. Okay? So iron and iodine are both examples, but there's many more that you guys have also discussed in your special assignment. Okay. So now we're going to just go ahead and do this fill in the blank activity about the pH scale. So do you guys remember what the pH scale means? What does pH stand for? Potential of what?
Hydrogen.
Hydrogen. Yeah, pH stands for potential of hydrogen. Okay, so that's what it's all about. And the pH scale ranges from what to what?
0 to 14.
Right, 0 to 14. Okay, things that are from 0 to 6 are what? Acids, right? Things that are 7 are what? Neutral. And solutions that range from 8 to 14 are basic or alkaline. Means the same thing. Basic and alkaline means the same thing. They're synonymous. Okay. So let's talk about these questions here. So letter A for number 4. A solution with a pH of 2 is an acidic solution. Okay, acidic. Number 4A. How about letter B? A solution with a pH of 9 is a basic solution. And letter C, a solution with a pH of 7 is a neutral solution. Letter D, in a neutral solution, the number of hydroxide ions equals the number of hydrogen ions, right? We're talking about a neutral solution, so that's going to just be equals. or is equal to whatever, same thing. How about with letter E? In an acidic solution, the number of hydrogen ions is
Greater.
Equal.
it's less. Okay? In an acidic solution, there's more hydrogen than hydroxide. So according to how this question is worded, you have to say the number of hydroxide ions is less than the number of hydrogen ions. Okay? So with letter F, how would we read that? In a basic solution, the number of hydroxide ions is more than, is greater than the number of hydrogen ions. Okay? Yes?
How do you know? I mean, are we looking at the pH scale itself, or?
Well, because remember, pH means potential of hydrogen, right? So when it all comes down to, you can describe a solution as being acidic, basic, or neutral by just its number on the pH scale. But when you look at that breakdown, the reason why there are those numbers in that scale, the reason why 8 to 14 is a base and why 0 to 6 is an acid, is because if you actually look at the solution in the beaker and you analyze its composition, and you got some hydrogens, okay, H+, and then you also have hydroxide in there as well. And if the concentration of that is equal, then that's neutral, right? You've got 3 hydrogens and 3 hydroxides. So that would be a neutral solution, okay? And there's a picture in the PowerPoint about it. But in this case, like for letter E, okay, here there's more hydroxide than hydrogen. So that's got to be a base, right? But if we reverse this and we kept that hydrogen and we got rid of this hydroxide, then that would be acidic. It's got more hydrogen than hydroxide. Okay, does that make sense? Okay, taking a look at that diagram on the next page there, and this is in fact the example that we talked about in class. So we see sodium giving its single outermost electron to chlorine, right? When it does that, you see that sodium takes on a positive charge, right? It becomes a cation ion, remember that, guys? Okay? And then the chlorine atom takes on an extra electron, so it becomes the anion, becomes negatively charged. And so that opposites attract, that positive sodium and the negative chloride ion are being held together by what kind of bond? Ionic bond. Okay? Ionic bond is the answer for number 7. Okay? How about number 8, a multiple choice question? The reactive properties or chemical behavior of an atom mostly depend on the number of, what's the correct answer? Is it the electrons in each electron shell?
Neutrons.
That's not the best answer, right?
Neutrons.
The neutrons, they're not important for that, right? No. It's option number 4, the electrons in the outer electron shell.
Is that called valence?
Those are the valence electrons, yeah. The number of valence electrons. The number of electrons in the outermost shell is the most important thing in determining the chemical reactivity of an atom. Okay, so for that multiple choice question for number 8, the correct answer is 4. The electrons in the outer electron shell of the atom, those are called valence electrons.
Does that affect the radioactivity as well?
No, the number of neutrons affects the radioactivity. Okay? Yes?
Can you explain number 7 in the ionic bonding?
Yeah. So basically, ionic bonding happens if an atom loses or gains an electron. Okay? So there's no sharing going on. So because that's another kind of bond, right? Covalent bonds are sharing electrons. So An ionic bond happens when one atom gives an electron to another. So the atom that loses the electron loses a negative charge, so it becomes more positive. We call that a cation ion. Okay? And then the atom that takes on the extra electron becomes more negative, and that's called an anion. Okay? And then so you have one positively charged atom and one negatively charged atom. So those are like oppositely charged like magnets, right? Opposite magnets. So they kind of are held together. They don't actually physically attach, but they are held together via those opposing charges. And that's an ionic bond, forming when an atom either loses or gains an electron.
Which one becomes the anion? Is it the chloride?
The one that becomes negative is the anion, and the one that becomes positive is the cation. Okay, I can put that on the board. So cation, positive, anion, negative. Okay. Now with number 9, what do you think is going to be happening there? We see fluorine. Fluorine has how many valence electrons in its outermost shell, guys? 7. And how many more electrons does it need to fill that second shell?
One.
Just one more, right? Because that shell is filled with 8 electrons. So it only needs one more. So that's very unstable. It wants to form a bond. Okay? Potassium, how many valence electrons does it have in its outermost shell? Only 1. Okay? So they're perfect partners, aren't they? So instead, the arrow is going to go the other way. Potassium is going to give its single most electron, the outermost shell, its valence electron, over to fluorine. Okay? So then potassium will become the cation, and fluorine will become the anion. Right? Potassium loses an electron, so it becomes positively charged. Fluorine gains an electron, so it becomes negatively charged, the anion. So now you've got 2 charged atoms. So what kind of bond's going to form between them? Ionic again. Ionic. Ionic happens when electrons are being donated and received, or gained and lost. Okay? The same example as with sodium and chlorine, except we're just going the other way with the arrow. Okay? You guys need to draw in an arrow on your paper showing the transfer of the potassium electron over to fluorine. Okay? Now with those 2 atoms of hydrogen and oxygen, let me just draw this out on the board really quick. So oxygen has 2 electrons on its innermost shell, that's full. And it has a second shell, and it's got 6. 1, 2 here, 1, 2 here, and here. Okay? So oxygen needs how many total electrons to achieve stability here? It needs 2 more, right? Because it only has 6 on its outermost shell. It needs 2 more. Well, luckily hydrogen is out there, right? So this is a hydrogen atom. I'm just going to put an H in the middle in the nucleus. Okay? Hydrogen, it's number 1 on the periodic table, so it's got 1 proton and 1 electron. Okay? So it only has 1 lonely electron in its outermost shell. The other hydrogen out there also only has 1 hydrogen on its outermost shell. How many electrons can hydrogen hold total on that 1 shell? Just 2. So how many more electrons does hydrogen need? They each need 1 more. So what's going to happen is that hydrogen is actually going to go and form an actual linkage bond with oxygen. Okay? It's going to do what? It's going to share an electron. So it moves over here and it joins up with that oxygen, and then this one also comes this way and shares that electron. Okay? I need to have this drawn a little bit better. Not the best artist in the world. Okay. And remember how that kind of gives that molecule kind of looks like a Mickey Mouse hat? It looked better in the image on the PowerPoint. I'm not a good artist, okay? But this is called what? What kind of a bond is this? You're sharing electrons. The sharing of electrons is a covalent bond. Okay? Specifically, what kind of a covalent bond is this? Polar. Yeah, this is a polar bond, polar covalent bond. Okay? Because the electrons are not being shared equally all around, right? There's more sharing going on this side of the atom than on this side. Okay, that's why it's called polar. Okay, that's more of a distinction you get into more in like a chemistry class. I more so just want you guys to know the difference between an ionic and a covalent bond. Okay, covalent bonds, electrons are shared. Ionic bonds, electrons are donated and received. Okay? All right, so number 10, a little bit of a math problem here, but not a very hard math problem. Okay, so we have an uncharged atom of argon. So it's uncharged. That means that it has not gained or lost what? It hasn't gained or lost electrons. It's uncharged. Okay, it has the same number of electrons then than it does protons. Okay, the normal number of electrons. Now we know two things here. We know the atomic number is 18, and we also know that the atomic mass number, the mass number is 40. Okay, but we have to figure out how many protons, neutrons, and electrons that atom of argon has. So the atomic number of 18 is a dead giveaway for at least one of the parts, right? What is the atomic number equal, guys? The number of protons. So we know right then and there that this atom has 18 protons. Okay? Now, being that it already said here that it was uncharged, you also know that it has how many electrons? 18 as well, right? If it's uncharged, it's going to have the same exact number of electrons as it does protons. Okay? So look at that. We can already go ahead and eliminate two of those options right there in that multiple choice question, right? B and D are out. Can't be. Yeah. Okay? So it's either A or C. A. You're jumping it up. Yeah, you're right. It is A. It is A. Okay? So why is that? The mass number is 40. What is the mass number? What is it the sum of? It's the sum of the protons and the neutrons. So all you have to really do is simple subtraction to figure out your number of neutrons, right? So you know that the mass number equals 40, and you know that the atomic number, which is just a number of protons, equals 18. So you just have to subtract 18 from 40, and that equals what? 22 neutrons. Okay. So it's choice A. 18 protons, 22 neutrons, 18 electrons. Okay. So just as a reminder, I'm going to put this up here on the board. So the atomic number is the number of protons, right? And then this number, the mass number, is the total of protons plus neutrons. So if you have that total of protons and neutrons, and you subtract out just the number of protons, you're left with the number of neutrons, right? Okay? What if you had to work backwards? Okay, what if you had to, this is actually more working backwards. What if you guys had to work forwards and I asked you this? An atom of carbon has 6 protons, 6 neutrons, and 6 electrons. What is the atomic number of carbon? 6. Don't get fooled up, okay? Okay, what if I said an atom of carbon has 6 protons, 6 neutrons, and 6 electrons? What is the mass number?
12.
12. 6 plus 6 equals 12. Okay? You could also just think about those questions. Am I asking for the atomic number, or am I asking for the mass? The mass number. Yes?
How did you say, what does uncharged mean?
Uncharged means it hasn't gained or lost electrons, right? The only way you get a charged atom is if you gain or lose electrons when you're forming an ionic bond. Okay? So because it's uncharged, that means it has the same exact number of electrons as it does protons.
question about
Sure.
the previous one.
Well, the previous one.
Yeah, the
Covalent
math number, no, the math number, the number 10.
Okay.
When an atom has, when the mass number is 40 and the atomic number is 18, the number of neutrons is 22,
Yeah.
right?
Yeah, that's right.
So is it a positive charge, negative charge, or no?
There's no charge. That's a neutron. It's an isotope. Okay. That's an isotope. So this example here is an atom that's an isotope, right? It has more neutrons than normal.
So it's radioactive.
Well, it could be. It depends. It depends. But so this, the normal atom of argon would have 18 protons, 18 neutrons, and 18 electrons. This atom is an isotope. You guys remember that? Isotopes are variations of atoms that have the same number of protons and electrons as they should, but they have a different number of neutrons. Okay? That's called an isotope. Okay, that was a good question. Okay, moving right along, guys. So we have this little easy diagram that kind of helps to summarize the different things we learned about in chapter 2. Okay, let me just clear up some space here. Okay, so this diagram A1, let me just go ahead and graph out. So this is A1 diagram, and so we have A, B, C, D, and E. Okay, so atoms have positively charged what's? What's letter A? What? Protons. Okay, letter B, atoms have neutral neutrons, right? Not a trick question, guys. Neutrons. Letter C, atoms have negatively charged electrons. Okay? Now letter D. So let's follow that sentence back to the beginning. Atoms have positively charged protons, and the number of protons present equals the atomic number. Okay. For letter E, atoms have neutral neutrons, and the number of neutrons may differ in isotopes. Awesome. Okay, that's what I was looking for there. The number of neutrons may differ in isotopes. Many people may have written mass number there, okay?
Yeah.
That wouldn't be totally incorrect, but more so that would be an extension of you would see maybe an extra dash coming off of isotopes, okay? You would say isotopes have different numbers of neutrons, okay? But the best answer that was being sought here for letter E was isotopes, but isotopes do differ in mass number, okay? Okay, so then following the C route, atoms have negatively charged electrons, and the number in the outermost shell determines the formation of, this line actually connects to the back of this, chemical bonds, okay? Chemical bonds. So now we're going to do A2. I'm going to go here. Okay, so diagram A2, and we need to fill in the blanks for G, F, E, F, G. Let's do that first. F, G, H, I. Okay. So, for letter G, chemical bonds. Electron sharing between atoms creates covalent bonds. Okay. Okay. And I'm sorry, going back to letter F though, so that one. So chemical bonds, electron transfer between atoms creates ions, and then the attraction between ions creates ionic bonds for letter F. Okay, ionic bonds. Right? Don't forget that difference, right? Covalent is sharing electrons, ionic bonds are donating and receiving electrons. Okay, so back to covalent bonds. Equal sharing gives you non bonds. But unequal sharing creates polar covalent bonds, just like in our water molecule, right? Okay? And it even tells you the example is water, and there you see your polar covalent bond. One side is more positive than the other, which is more negative. Okay? And then there's two different paths here to get to I. So water has important qualities due to polarity and this, and then the polar covalent bonds can lead to these three little dots between these water molecules. What kind of a bond forms between water molecules, guys? Hydrogen bonds. We're looking for hydrogen bonds here.
What's the definition on non-polar?
Equal sharing of electrons. Equal sharing of electrons all around. So like an example of a molecule that is bound together with as a non-polar covalent one is methane. Because with methane you've got your central carbon atom, and then you've got an equal spread of hydrogens around that. That would be a non-polar covalent bond. Okay? All right, look at the erasers again. You see that? That's non-polar. You've got an even distribution and sharing of the electrons all around the central atom. Whereas polar covalent, it's a bit offsides. It's unequal. Okay? So that's that diagram. So we are on the last page. Okay? Do you guys want to go over this organelles checklist first, or do you want to just go in order and do this? The chapter 1 easy stuff.
Yeah.
You want to do what? You want to do the organelles? Let's have a vote. Who wants to do the organelles now? Okay. All right, fine. That's not fun. The other stuff's easy stuff, right? Kind of. Okay. Let me spare some room here. We're going to do all of it, of course. Okay. All right, guys. Number 1. Just before we start going over these answers, though, I just want you guys to know that there was never a separate rough or smooth ER here. One of them is just going to be ER, endoplasmic reticulum. All of the functions are just smushed into one. And that may have been something that kind of threw you off a little bit because you were looking for rough and smooth. But one of them, the answer is just endoplasmic reticulum. Okay, it's a very general thing. You'll see it. Okay, that's the one thing I don't like about this. I didn't make this worksheet, obviously. Okay, but I like it for a lot of other reasons. Okay, so number 1 controls the movement into and out of the cell. What's that? The cell membrane. Okay, so what controls movement in and out of the cell? The cell membrane. Okay, so that's number 1. Number 2, the watery material, which contains many of the materials involved in cell metabolism.
Golgi?
Golgi is a post office, right? The cytoplasm. The cytoplasm. It's mostly water. When you're in lab and you look at it under the microscope, it looks like nothing. It's mostly water. Okay? Number 3, lots of different jobs here. Serves as a pathway for the transport of materials throughout the cell, also associated with synthesis, which means it makes stuff, and it also stores stuff, okay? Synthesis and storage. So what's going to go there? Does a lot of things. It's the ER, endoplasmic reticulum. Okay? Endoplasmic reticulum for number 3.
And is that smooth or?
It's all combined in that one. It's not differentiated in this worksheet. Number 4, serves as the control center for cell metabolism and reproduction. The key word is control center. The nucleus. Nucleus. Okay, number 5, protein synthesis. What organelle assembles proteins? They happen, these organelles are found on the surface of the rough ER. Ribosomes. Ribosomes. Yes, the rough ER is involved, but specifically what organelle doesn't, the ribosomes.
Does that pass through the nuclear envelope and attach to the ER?
Yeah, exactly. Okay, number 6. Involved in the digestion of food within the cell. Okay.
Lysosomes, right?
Yeah, lysosomes. Digestion is the key there for lysosomes. Number 7, the powerhouse of the cell. It makes ATP. Yeah, mitochondria. That's what does cellular respiration. That's our powerhouse. Mitochondria. Number 8, packages and secretes the products of the cell. Golgi. You guys are all so quick on that Golgi apparatus. Golgi apparatus. Number 9, involved in cell division in animal cells. Well, they are involved, but specifically what is needed to divide the chromosomes. It's called centrosomes. Centrosomes with centrioles. Okay? Centrosomes with centrioles.
What's an example when it's without?
Plant cells don't have centrioles. That's one of the differences between plant and animal cells. Animal cells have centrosomes with centrioles. Plant cells just have centrosomes, no centrioles. Okay. Number 10, fluid-filled organelles enclosed by a membrane, and they can contain stored food or stored waste. There's many different kinds of them. Vacuole. Vacuole, right? There's food vacuoles, there's waste vacuoles. But the answer there is vacuole. Vacuoles, plural. Okay. Number 11, the site of the production of ribosomes. Where are ribosomes made? The nucleolus. Yeah. Nucleolus. Number 12, controls movement into and out of the nucleus. The nuclear envelope. Or the nuclear membrane, it means the same thing. Nuclear envelope / membrane. Okay, let me get rid of this. Okay, 13. Gives the cell its shape and provides protection not found in animal cells. Cell wall. And number 14, hair-like structures with the capacity for movement. They are short and numerous. Cilia.
Is that found in the lungs?
Yeah, well, it's found in many different places. In us, we have it in our lungs. In organisms like paramecium, it surrounds the exterior of the cell. Okay, cilia. Number 15, a long hair-like structure used for movement. Flagellum. Okay, so this is like using the singular form of the word, which is flagellum. But if you're talking about the plural form of the word, it's flagella. Okay, flagello plural, flagellum singular. Number 16, the site of photosynthesis. What is the energy converting organelle in a plant that does photosynthesis? The chloroplast. The chloroplast. Good. And number 17, we are omitting for right now. You will learn about it once we get to chapter 8, but no need for that right now. Number 18, rod-shaped bodies that carry genetic information. No, that was the chromosomes. Chromosomes, which are made of chromatin. The chromosomes. Rod-shaped bodies that carry your genetic information.
And that becomes both DNA and RNA?
DNA is on the chromosomes. For some organisms, they don't even have DNA, they just have RNA. So some very simple organisms and viruses, for example, are just RNA.
That's
They don't even
prokaryotic.
have DNA.
Prokaryotic.
Viruses are not prokaryotic or eukaryotic, they don't have cell structure. They're weird, they're really not living things.
Okay.
What is the phospholipid membrane?
The phospholipid bilayer is that's going to be the component of the cell membrane. Cell membranes have a phospholipid bilayer where they have hydrophilic heads that are in contact with water inside the cell and outside the cell, and then hydrophobic tails. That's the bilayer. Hydrophilic heads and hydrophobic tails.
So would that be an acceptable answer if you put phospholipid membrane or just
If you put phospholipid bilayer, not really, because the cell membrane also includes proteins that are embedded in the phospholipid bilayer. So a cell membrane is both phospholipids and proteins. Okay, so that's why cell membrane is the best answer. Okay. All right, we still have the chapter 1 questions to go over, and then we're done. Okay. So let's see, guys. Difference between population, community, and ecosystem. You guys remember that? Okay, so a population consists of what?
Individuals.
Okay, so same species, that's the key. A population is all the individuals of same species that live in the same place at the same time. A community is all the populations of different species, right? All pops of different species. Different species, okay? And an ecosystem, yeah, all the communities of living things as well as the non-living abiotic factors. Good. So all living and non-living things. Okay, that live and interact at the same place at the same time. Okay, so remember that. It's a fancy way of saying living things is biotic, and that the non-living things are abiotic. Okay? So that's important differences to be aware of. Populations, all the same species, community, all the different species, and then ecosystem is all the different communities of the species, plus the abiotic factors. Okay. Next question. Describe the hierarchy of life as it increases in complexity. So where do we start? Where can we start? What have we talked about, right? DNA is what? What is DNA? It's not a cell, right? It's a molecule within a cell. So when you talk about the hierarchy of life as it increases, right? We go molecule, and then what's next? Getting bigger. Cell, okay? And cells make up tissues.
Organelles.
Okay. Well, actually, organelles are going to be inside cells. So actually, organelle can go here. Okay. But working our way up, tissues make up what? Organs. And organs make up organ systems. And organ systems all together make an individual organism. Okay? Organism. Okay? And so an organism, many organisms together of the same species are what? Now we're into this part of our hierarchy, right? Population. And then what's next? What's bigger than a population? Community. What's bigger than a community? Ecosystem. And what's the biggest?
Biosphere.
A biosphere. Yep, that's the hierarchy of life as it increases in complexity, from small to large. Okay? List at least 3 properties that unify life on Earth. There was, I think, 7 that we went over. If you saw a question like that, could you give me 3? What are some of those properties, guys? Okay, so order, right? There's an order to life, and this is the order of life, right? Order. What else? You don't have to give it to me in order. But what else? What are all organisms on Earth capable of? Okay, growth and development, reproduction. Growth and development, reproduction. Okay, we can keep on going, right? Response to the environment, energy processing, evolutionary adaptation. Okay, there were several things that we talked about here. But if you saw a question like that, you just have to list 3. Okay. Moving on, the mechanism for evolution as described by who is what?
Charles Darwin.
Yeah, Darwin, and the mechanism is natural selection. Okay, fantastic. That's as opposed to artificial selection by humans. Like for example, you know all the different dog breeds that exist? All of those dog breeds came from the wolf as the common ancestor, and humans selected tame wolves, and that's how they got their pets, right? And then from those tame wolves, they selected for different behaviors and abilities. Like some are really good at killing rats, the ratters, the terriers became that group. Some are really good herders, so they can help you out there herding sheep, like the German shepherds and the collies in that group, okay? So there's all sorts of different breeds of dogs because of artificial selection by humans. It was not imposed by the natural environment. Okay? It was based on what humans wanted in their animals, their domesticated animals. Okay, number 5, list at least 3 abiotic factors that may impact diversity and abundance of life and indicate whether each factor flows one way or is recycled. Is that a tough question?
No.
Okay. For some, yes, for some, no. All right, give me some abiotic factors.
Air, water, temperature.
Okay, water. I like that. Temperature. Where does the temperature come from?
The sun.
The sunlight. So sun's energy.
Sun and wind.
Sunlight, which is energy. Okay. Give me one more. Okay, like nutrients. Or you said air, so we can say like carbon dioxide, right? We can say carbon. Carbon as in the carbon dioxide in the air, okay? So the sunlight, do you guys remember, this was in chapter 1, there was a little graph. What happens with energy in the environment? Does it get recycled back to the sun, or does it flow one way? It flows one way. So the energy flows in a one-way direction, gradually being lost through each transfer, okay? What about water? Does water flow one way, or does it get recycled? It gets recycled. Yeah, it recycles. And what do you think about carbon and the other chemical nutrients? Chemical nutrients, do they flow one way, or do they recycle? They recycle, okay? And I know, guys, those of you who are in my environmental science class, these are really easy questions for you. But those who are in just bio, we hardly touched on this in chapter 1. Okay, but it is an important thing. When we have a more of a regular term as opposed to a blended term, we have more time to get into all that. Okay, but I do get into that a whole lot deeper in environmental science. Okay, so those are some of the things that recycle versus flows one way. Okay, 2 more questions left, guys, and then we're done. So what are the steps of the scientific method? You guys remember? Okay, so what is the very first step of the scientific method? You make an observation and you ask the question, right? You see something interesting, you're like, why is that happening? Okay? So you make observation, ask a question. Very good. You formulate your hypothesis, right? Your educated guess. You formulate hypothesis. Once you have formulated hypothesis, what are you going to do? You're going to test the hypothesis using what? Your experiment. Okay, so you're going to design and perform experiments.
need to have a placebo group involved in that as
A
well?
what?
A placebo?
Well, that would be part of your experiments. You have your control group, your placebo group, and then you have your experiments groups. Sure. Okay. So so once you analyze your data, you're going to draw your conclusions. Okay. So draw conclusions. And is that where it's going to end?
No.
What are you going to have to do then? Share it. You're going to share it so that people know about what you found. Share info. So you can do that by presenting, you can do it by publishing, you got to share it so that you can share with the scientific community and the rest of the world what you found. Okay? So that's just, a rough sketch of the scientific method. Now, in terms of the hypothesis versus a theory, we went over that in that chapter 1 lecture, right? Remember? Okay? So a hypothesis, so first of all, what is a hypothesis?
Educated.
It's an educated guess, okay? Has it already been tested or does it need to be tested?
Needs to be.
Yeah, needs to be tested. So a hypothesis is an educated guess that needs to be tested, right? Using an experiment so you can carry on with the rest of the steps. What about a theory? How does something become a theory? It's a well-tested hypothesis that's supported with a giant amount of evidence, but we don't use the word proven, okay? Proof is an ugly word in science, okay? A well-tested hypothesis that has never been rejected. It has always been supported. And therefore, because it's supported by so much evidence, it is commonly accepted as true by the scientific community. But we should never use the word proof. Because if the technology changes, maybe that will allow us to study the world a little bit differently. Okay? So we always will use the words reject or support when talking about the outcome of a hypothesis, okay? And a theory only happens when a hypothesis has undergone rigorous testing over and over and over and over again. It's never been rejected. It has always been supported, okay? And this has happened, and it gets tested by scientists all around the world over many years. It takes a long time for something to become a scientific